Hello, my name is Michael Reidenbaugh, and today I'm going to be discussing research that I performed in my molecular biology course. The goal of the research was to isolate bacterial species in acid mine drainage that may be able to be used in passive remediation treatment. So some background, acid mine drainage is runoff from abandoned mines that will contaminate fresh water. This makes the water uninhabitable to most forms of life, except some bacteria, and there's no way of fixing it. You can only treat it. Passive remediation is one of the methods of treatment to reduce the damage that has been done to the environment. Bacteria have a significant role in passive remediation, in which they will precipitate metals so that the metals become solid and can then be filtered out of the water. So to start my experiment, what I did was I took a sample of acid mine drainage and serially diluted it so that I could form spread plates. And the purpose of what that was to obtain isolated colonies. And as you can see in figure 1, there is a high diversity in the different types of colonies. You can look at the shape and colors, they're all different. And this is interesting because it used to be thought that nothing could live in acid mine drainage, and it was just too extreme. Nonetheless, I did get a variety. I isolated 2 colonies. You can see what they look like in figure 2. And so what was done next is that DNA was extracted from each species, both isolates, and they were run through PCR to make multiple copies. The DNA just being the bacterial genome. This was done so that the genomes could be sent out to a company. I couldn't perform this, but the company performed full genome sequencing so that the isolates could be described. You can kind of see how this is done in figure 3. I'll explain it. So the DNA is broken up into small fragments, and overlaps in these fragments are used to construct larger fragments, these being called contigs. And the contigs are of high accuracy. You're basically almost guaranteed that that is what the actual sequence will look like. And gaps between each of the contigs is where it's unsure whether or not that's where the contigs will overlap. So there are differences in the level of accuracy. So contigs are high accuracy. Moving back up to the full genome, it's lower accuracy. Nonetheless, from the contigs, I was able to see a variety of metal resistance genes. And there are full operons for copper, cadmium, cobalt, and zinc resistances. I also searched for antibiotic resistance genes, and out of the more common resistance genes, only macrolide resistance was found. What was done next is I attempted to find out what the organism actually was. And through the main program that I used, there was no real identity. You can see in figure 4 that it is related to species in the Cupriavidus genus. And so to actually see if this is a novel bacteria or not, I ran the 16S ribosome sequence through the BLAST algorithm, which is on a website. And this actually showed that the organism that my partner isolated was called Cupriavidus pauculus. And this is kind of supported by the species tree, since it shows that they're from the same genus. Nonetheless, Cupriavidus pauculus is not actually well known for having metal resistances. It's really only known to cause infections in humans. It's actually quite interesting because looking into it, it's known to have multiple antibiotic resistances, and whenever I searched for antibiotic resistance genes in the organism, not much was found. This might just be because I searched for common ones and not for every single antibiotic resistance genes, but it This result might also be because the antibiotic and metal resistance could be because of shared mechanisms. So for example, efflux pumps that might pump out metals as well as antibiotic compounds. Overall, this is a significant finding because there was an organism that was isolated from acid mine drainage, and it has multiple complete operons for metal resistance. So for future testing, more can be done to understand the specific mechanism of the metal resistances in the isolates. And if there is a lot of precipitation being done, then it would be interesting to determine if the species would be promising for passive remediation. Thank you for listening.